Welcome back to An Ape's Guide to Human Language. In this course, we are taking the perspective of a curious ape, trying to figure out what it is about human communication that makes it so different from other species in our clade. So far, we've talked about a lot of features of the vocal production anatomy and a lot of features of the anatomy outside of just the vocal production organs. And we've started to settle on this idea that maybe there might be something special about our brains. In this module of the course, we're going to be looking in particular at the great brain expansion that seems to have gone on in the most recent stages of human evolution. We've taken a look at this diagram in the past about the rate of cranial expansion that has been observed across the fossil record within our clade. And we can see that although brain expansion progressed rather gradually for a very long period of time, something strange happened at around 1.8 million years, where there is a rather radical uptick in the rate of cranial expansion. And that's what we're really interested in right now. When we looked at this data before, one of the things we had to take into account was our activity budget. There are certain things that a primate has to do simply to stay alive, and our core activity budget comprises the time spent feeding, the time spent moving, some of which is necessary in order to acquire food, and enforced rest, like sleep or not being able to move due to climate variables. Once we have taken into account all of those constraints upon our time, we have what's called free rest left over. And free rest is the time that we can spend doing things like socializing, one of the predictors of our community size. We can see from this visualization here that in order to maintain a brain and a body the size of all of these different primates in our lineage, it takes a certain amount of time simply for chewing and digesting the food stuff that we need to power our brains and bodies. So we can see in this diagram that pan troglodytes, that's chimpanzees, are eating for about 8 hours a day. That's a full-time job. We can see the gorillas are eating for something like 8.5 to 9 hours a day, which is overtime if you think about it. And they need to eat that much just to amass their gigantic body size. When we look at those few examples of Homo erectus, Homo heidelbergensis, Homo sapiens, and Homo neanderthalensis, we would need to be spending more than 9 hours a day eating in order to sustain a body and a brain the size that we have. And that's just not feasible in our core activity budget. So, as we've discussed previously, we have to do something to get more energy out of our foods, and we have to be able to do that faster. We've talked before about the idea of the seas. We might be able to utilize a bunch of different strategies to get more energy out of our foods in a shorter amount of time. We can cut and crush our foods as a form of pre-digestion, which reduces the amount of time we have to spend chewing in order to digest the caloric content of raw foods. We can also carry foods from one place to another, which reduces the amount of time we have to spend moving from one place to another simply to obtain that nutrition. It's worth thinking about that carrying property from the perspective of our broader anatomical substrates. We're able to carry quite a lot of stuff, given that we have two free limbs that are not obligatorily used for locomotion. So our bipedalism gives us an advantage in being able to process and move foods from one spot to another without having to compromise our mobility in so doing. The idea that we've heard about already in a bit more detail is that cooking might be critically involved in helping us to get more nutrition out of our foods. Finally, another process that sometimes gets forgotten is if there are foods that we're eating a lot and we're transporting those foods with us as we go, there's a high likelihood that the most delicious of those foods might end up being encouraged to grow in more delicious forms. Uh, it's unclear whether early cultivation started as an accident of what seeds got dropped or deposited in high nutrition contexts that allowed them to grow and become productive. And this may have been an accidental process, at least to begin with. But at some stage, our early ancestors did indeed have some intentionality in helping some plants survive more than others. And this has also led to higher nutrition density in some of the foods that we eat. For example, if we look at the wild type of a carrot, it's a weedy little root that doesn't have much food on it. But when we look at cultivated carrots that we can buy in a grocery store, they're big and juicy, and plucking just one carrot can actually give you a much more nutritious meal than a wild type carrot root. So now that we're thinking about food in our evolutionary history, we can understand a bit more about how our brains got so massive compared to everybody else's. Over the next few slides, we're going to be looking into which parts of our brains got the most massive. So in this slide, we can see a cladogram of mammals. At the top on the left, we can see the mammalian common ancestor brain, and at the top on the right, we can see the primate common ancestor brain. And even just looking at those two examples, we can see some radical differences. We see a small difference in size between the mammalian common ancestor and the primate common ancestor, but more importantly, we see that one of these brains has a large overall grey patina, the primate brain, versus the mammalian common ancestor, which is much more colourful. So what we're actually looking at in this visualization is the distribution of different functional areas in the brains of different mammals. And as we work our way through that mammalian family tree, one of the things that should strike us is that the primates, in particular, the large brain primates like the chimpanzee and the human, have a much, much larger area of the brain that is shaded in that gray color. So we've zoomed in now on just a few examples, and what we see is that the blue colored areas of these different brains are those areas that are primary and secondary visual areas of the brain. That means there are nerve signals from the eyes that are being transported right to the back of the brain, where they are processed for the visual information that they contain as sensory areas. So primary and secondary sensory areas are colored in blue. And we can see in the mammalian common ancestor, that blue part is in a slightly different location than in the primates. But even between the chimpanzee and the human, we can see that the primary and secondary visual cortex are taking up a smaller proportion of the cortex in the human than they are in the chimpanzee, and a smaller proportion in the chimpanzee than in the primate common ancestor. Next, if we look at the red-colored area, that red is indicating for us the primary somatosensory area. So that's the headband across the top of the brain. Now, the somatosensory area is the part of the cortex that is bringing in nerve sensations from the rest of the body. This is the part of the brain that is very, very helpful for figuring out where our bodies are in space and what are the sensations that we are experiencing in our bodies. The primary somatosensory cortex is just behind the motor cortex, which is where we send out signals for how we're going to control the muscular functions of our body. So the motor cortex is right in front, and just behind it is the somatosensory cortex. So we can understand quite clearly that when we send a signal from the brain to control the body, we then get some feedback into the somatosensory cortex, telling our brain whether or not our body has done the instruction that we asked it to do. What we observe, of course, just like for the primary and secondary visual area, this somatosensory cortex is taking up a much, much smaller proportion of our cortical tissue in humans as compared to chimpanzees, and in chimpanzees compared to our primate common ancestor, and in our primate common ancestor compared to our mammalian common ancestor. When we look to the yellow and orange regions of the brain, these are the primary and secondary auditory areas. So now what we're able to see is that when auditory information comes in through the ears, it gets passed up to the primary auditory areas for processing of the sounds that we're hearing around us. And from there, it's passed to the secondary auditory areas where it is further processed for its information content. So once again, we're seeing a sensory area that is a lot smaller in the human brain as a proportion of the overall cortex as compared to the chimpanzee brain, and smaller in the chimpanzee than in the primate common ancestor, and smaller in the common ancestor than in the mammalian common ancestor. Now, you might be wondering, does the size of the cortical area really connect to functional relationships in the way an animal moves around in its environment or lives its life? So it's important for us not just to speculate that a larger area means that a larger proportion of cognition is devoted to sensory processing, but also to check for examples where that might be clearly laid out for us. Now, on this slide, we have a few beautiful examples of some animals with very specialized sensory organs. So we have a duck-billed platypus up on the top left. And what's interesting about the duck-billed platypus is that it has in its bill some electrosensitive hairs that help it when it is hunting underwater to find small organisms that it finds particularly delicious. So when we look at the shape and structure of its sensory cortices, it has very large areas that are devoted to particular kinds of sensory receptors. And their organization and shape is quite different to other animals. When we look at the star-nosed mole, which has a very interesting nose shape with lots of very sensitive tissue structures on the very end of it, it also has a very unusual shape for the cortical representation of the sensory information that is going into its nose. At the top right, we can see an example of a raccoon paw, which has 5 separated digits that work quite like hands. And one of the things that's interesting compared to others in its clade is that it has a sort of a star-shaped representation for the individual digits separated out in the sensory cortex, which is quite different from others in its clade who have more of a paw representation where the fingers are not so distal and can't be represented quite so separately. And then finally, with the naked mole rat, we can see that it has a quite unusual form of sensory input as well, with different kinds of sensitivity being reflected in different structures in its sensory cortices. So this should give us some reason to be confident that the size of a particular sensory area on the cortex reflects how much processing is being done for that sensory modality as compared to the other jobs the brain is able to get on with. We're going to look at one further example of how we might understand this data. And this time we're going to go back to our friends, the glides. So this is the next door neighbor branch of our mammalian family tree. So now when we compare the brain of a capybara to the brain of a squirrel monkey, What we can see is that the capybara, even though it has objectively a larger body and objectively a larger brain, the vast majority of its brain cortical area is taken up by those primary sensory areas. By comparison, the squirrel monkey, even though it's much, much smaller, both in body size and in brain size, the squirrel monkey has a much smaller proportion of its cortical surface taken up by those sensory jobs. So, what is going on between these two animals? How is one of them requiring such large cortical areas and the other only requiring a little bit of cortical tissue to do the same job? Is it that the squirrel monkey doesn't really sense its environment as effectively or truly or deeply as the capybara? Well, we have to remember our primate brain scaling rules. So when we were looking at this previously, it had become clear from the work of Susanna Herculano-Halsall that the density of different brains really matters here. What we see across the mammalian clade in general is that the larger your body, the larger your brain, and the larger your brain, the larger your neurons, which means that ultimately you can have a bigger brain, but lower neuron density in a bigger brain. So that capybara, if we do the sums and estimate out the density of neurons in half a microgram of capybara brain, it would come out at something like 11 neurons per half microgram of brain. By contrast, the squirrel monkey, even though it's got a much tinier brain, in the same half microgram of brain would have 43 neurons. That means the squirrel monkey is able to pack those cortical neurons in much, much, much more densely, which means that not only do the sensory areas take up a smaller proportion of the cortex, but the cortex itself is like thriving with densely packed neurons. So what are we doing with all of these densely packed neurons that are not obligatory for our sensory processing, that are not required for us simply to understand how we experience the world? Well, this is a great time for us to think about associative learning once again. So associative learning is that process by which humans and animals form a link between two mental representations. We've seen a beautiful graphical representation of this from the graphic novel Neurocomic. But it's worth also remembering the two processes that are involved in this kind of learning. First of all, we have that process of Hebbian learning, which we can simplify as neurons that fire together, wire together. And if you've taken cognitive psychology already, you'll be familiar with this concept. If you're still yet to take that course, it'll be coming up for you next semester. The second process that we can also highlight is long-term potentiation. And this is a change that occurs at the synapse that changes how frequently a particular cell is likely to fire in response to a particular kind of input. So those two processes, Hebbian learning and long-term potentiation, are the source of forming connections between neural representations of things. I also like to highlight, though, that the brain is made of more than just neurons. So in this visualization, we have an introduction to brain cells of different kinds. And what we can see curving through the middle of the illustration is a big old neuron with a big head surrounded by a gorgeous dendritic arbor. Those dendrites are where the neuron is going to receive incoming signals from its own synapses. And if it accumulates enough signals through the dendritic arbor that an action potential is triggered, then just beneath its chin at the axon hillock, a signal will be transmitted all the way down its long axon to the axon terminal buttons, and this is how our neural signaling progresses. Now, it doesn't often make it into the introductory textbooks, but it's worth us thinking about the ways in which information is adaptively processed and how much the brain is changing all the time. So we have illustrated here in the nice round-headed cartoon characters the oligodendrocytes. And oligodendrocytes are a form of astrocyte, which is one of the non-neuronal brain cells. And the oligodendrocytes, they have little arms known as processors, and their processors will wrap around the axons of any neuron that it's firing. And the more frequently it fires, the more they will wrap their little arms around. And it is the wrapping of the processes of the oligodendrocytes that create the myelin sheath that helps the neuron to transmit its signals faster. So here we go. If the oligodendrocytes are actively choosing to wrap their processes around an active axon, that means the more frequently a neuron fires, the more efficient that neural transmission pathway will become. And as for a neuron that does not fire very often, that neuron will eventually start to have the oligodendrocytes unwrap from the myelin sheath and potentially send their processes off in a different direction to serve a different neuron. This is real use it or lose it stuff. In addition to the oligodendrocytes, we can see a bunch of other different kinds of astrocytes that are involved in regulating processes and transmitting nutrients to axons and regulating activity at the synapse and doing all sorts of other jobs as well. But my main point in sharing this idea of the brain as an active, living entity, not just made of electrical wires that sometimes fire and sometimes don't, is to remind us that there is a vast potential for changing the states between those neurons and the structural organization of the brain. So, with all of these different learning processes and all of these different ways of changing the way that information is represented or connected in the brain, does this mean that humans are somehow superpowered associative learners? Is all that free neural tissue, the tissue that is not in the sensory areas and therefore has a predefined job of understanding what's coming in through the eyes or understanding what's coming in through the ears or understanding what the body is feeling at any point in time, is all of that free neurological tissue maximizing our capacity to learn kind of anything? Is that what's going on here? Let's take a look at how brains are connected in ways that might help us answer that question. So this is an interesting study that has looked at the strength of functional connectivity of brain regions depending on how nearby or how far apart they are. Now you'll notice that the image of the brain here is a little bit different than the images that we've been looking at before. And that's because this particular kind of visualization has taken the sort of wrinkly surface of the cortex of the brain and kind of inflated it so that we can see a little bit more clearly all of the area rather than having rather a lot of it hidden inside those hills and valleys. So when we look at this visualization of the brain, we have the blue color representing areas of the cortex that have stronger functional connectivity for neurons that are right next door to where they are. When we look at areas that are yellow, we're looking at areas of the brain where the majority of the neurons have stronger functional connectivity for areas of the brain that are far away. Now, what's interesting when we look at this visualization of the brain is we can already see some areas that we've just been looking at before. That back part of the brain, the occipital cortex, which is where we saw the primary and secondary visual cortices, is colored in blue. And what that's telling us is that the neurons in the visual cortex, in the primary visual cortex, are mainly communicating with the next door neighbor neurons in the visual cortex. That's literally how the visual cortex works. We take information from the retina and we pump it into visual cortex, and then there is a whole pile of processing that's just trying to understand how the different nerve signals in the retina could be related to one another in order to tell us what we're seeing. So we see in the primary visual cortex the greatest strength of connectivity for neurons that are communicating with those neurons that are very, very near to them. When we look further forward in the cortical tissue, we can see once again that headband. We can see the somatosensory cortex. And I mentioned before that there's the motor cortex that is just in front of the somatosensory cortex. And we can see those two ridges in the cortical surface, the motor cortex slightly towards the front, and then the somatosensory cortex right behind it. And those areas also are regions where the highest functional connectivity, the highest strength of connections between neurons is observed for neurons that are right next to each other. Now, this one makes a great deal of sense. If I'm tracking nerve inputs on my hand, the neurons that are right here on my hand that might be activated when I touch it are going to be active at the same time as the neurons that are right here. I moved my finger just about 1 millimeter, but my finger is bigger than a millimeter. So a lot of the same neurons were active when I touched in those two locations. So once again, we see the highest functional connectivity for neurons that are right next door to each other, because when we sense something happening in our body, we're likely to be sensing something similar in the next door neighbor region of our body. In the motor cortex, it also makes a certain amount of sense. If we're sending nerve signals to control some part of our body, like our hand, there might be lots and lots of representations for different parts of the hand that are coactive simultaneously for the motor control of that body part. So once again, we're seeing sensory cortices and motor cortex are showing highest functional connectivity for next door neighbor regions. If you remember, we also saw those primary and secondary auditory cortices just down the side at the very top of the temporal lobe and the temporoparietal junction, just in here. And we can see again on this brain, those areas are also most likely to be talking to next door neighbor regions. And this has to do with the fact that we represent certain kinds of sounds that are more similar to each other in their frequencies in brain regions that are very close to one another, because we're likely to experience co-activation of similar frequencies. When we're hearing one, we're likely to be hearing another that's quite similar to it. So what we are seeing then is the idea that the non-primary sensory cortices are regions of the brain that have a higher proportion of their connections to very distant brain regions. Now, that's an exciting prospect for us, because what that's telling us is that we're able to coordinate or learn associations between things that are very different from one another. So if we are expanding the parts of the brain that are not just about sensation, then we're expanding our capacity for random connectivity patterns. We're expanding our capacity to learn anything, which is a really exciting prospect. In this second visualization, what we're looking at is a visualization that, rather than being taken from the average of a large group of people who are all evaluated for their average connectivity profile across their brain, the second visualization is looking at how similar versus how variable different individuals in the scanning set were. Now, the first thing that we see is when we look at the motor and somatosensory cortex, we're seeing that most people are very similar to one another. That blue color in this case indicates that the neural representations, in terms of functional connectivity, are more similar in those regions. Across the headband of the brain, we're seeing high similarity, And we can understand this quite clearly as the idea that most of us have very similar body plans. Now, obviously, that's not going to be 100% of the population, but for those of us who have two arms, two legs, one body in between, we're going to have quite a lot of overlap in how our senses are wired into those brain regions. We see less similarity in the visual cortex, and this can be understood in terms of the idea that we all have to learn how to see, um, from living in the world, opening our eyes, and figuring out what is a meaningful visual stimulation. It's entirely possible that different humans do that in slightly different ways to one another. When we look to the auditory cortex, there's relatively high similarity once again. And this is indicating to us that the structure of our ears is very similar from one person to another, and that the way we process auditory information tends to be so too. When we look at those other unrestricted brain regions, what we're seeing is more variability. And this ties in with the idea we were talking about before, where they are connected to more distant brain regions, which means we can connect up more variable things. And that seems to be the case. Each individual is learning to connect up different kind of signals in brain regions that are far away, but in patterns that are different from one person to another. This is a great way of thinking about experience-dependent learning. The idea that we have to live in the world to acquire knowledge. This is not just instinctive or preloaded software. This is like setting up an operating system and then letting it run wild while we learn about our environments and how things work within our context. It's also interesting to note that these are the brain regions that are kind of understood to mature latest in human brains. The frontal cortex and, of course, the temporal cortices, which continue developing even into older age. So our primate might want to take another look and think about how they fit into this primate family tree of brain development. What does my association cortex look like? So here's our cladogram once again, and this time around we're going to zoom in on the macaque brain. This time around, we're going to look at a similar kind of analysis. It's a functional connectivity analysis, and this time, instead of looking at the difference between individuals within one species, which we were looking at before, how different is one human to another, this time we're going to look at interspecies difference. So remember, this is a macaque. They're not super closely related to us, but they are primates. They do have two arms and two legs. Let's see how that goes. So when we look at this macaque brain, what's interesting is we can see a huge similarity between one species and another. When we look at the motor and somatosensory cortex, mainly the motor cortex, to be fair, we see a lot of similarity between macaques and humans. This means controlling our bodies is done in largely a similar way. When we look at the back of the brain and the visual cortex, that blue area, once again, we see that on average, the overall macaque pattern of visual processing is very similar to the overall average of human visual processing. The functional connectivity patterns are very, very similar for the primary and secondary visual cortex and for the motor or somatosensory cortex across the species divide. We can also see some similarities in auditory processing as well. But where we see the biggest differences between humans and macaques is in those long-range connection areas, those areas that are more variable from one human to another, and those areas that are later maturing in humans. So, our curious primate might be thinking, my brain is most similar to humans in the primary and secondary sensory cortices, which are relatively small as a proportion of my overall brain tissue. But they are a bit bigger in me compared to the amount of cortical tissue they take up in humans. So what is it then? What is it that humans do with all this excess brain power? What is it that they do with this massive association cortex? And that's a question that we will be returning to in our upcoming content. See you next time.